I will start. Quitter la ligne. J'ai compris. Je peux dire, c'est ça. C'est ça. Ça va, ça va. Today I'm talking about polymer-ceramic composite coating on biodegradable magnesium for biomedical implant, especially orthopedic implant. You see, we are using polymer, ceramic, magnesium is a metal, coating. First of all, we have to get information about what do we mean by coating. Coating is a way just to tailor the surface of a material. Our material today is magnesium. So coating is to tailor or control the surface of this material for a certain applications by inducing some other properties, which might be electrical, mechanical, chemical, or biomedical or functional properties. So coating is very important to tailor the functionality of magnesium. First of all, we have to get information about material. What do we mean by material? Polymer is a material. Metal is a material. Ceramic is a material. Metal like steel or magnesium, which is in our target today. Polymer, clothes, nylon, plastics. All plastics are polymer, but not all polymer are plastics. We have two kinds of polymer, synthetic polymer and natural polymer. Natural polymer like silk, wool, chitosan, which is also included in our work today. Collagen is a natural polymer. And then the third class of material is ceramics. Ceramics, pottery, our sanitary ware in our home, for example. All these kinds are ceramic. We call it conventional ceramic because it's not very pure. There is also another type of ceramics, we call it technical ceramic. Technical ceramic, we call it ultra pure materials for specific application. We have to induce some functionality. We have to use technical ceramic, which is also bioceramic, is technical ceramic, but for bio applications. The first classes, which is a composite. Composite is not a compound. Compound, we call it like sodium chloride, whatever, is a compound, but not composite. Composite, which means you have two or more different material having different chemical and physical properties completely, put them together or merge them together to get another superior product with different properties. That means you could bring, for example, polymer and ceramic. Both have different chemical and physical properties. Put them together to get another properties, which could be used for which application you are planning to do, for example. Our application is implants. That's the market share by 2021. Mainly joint reconstruction is the highest. And then spinal implant, trauma implant, there is also other application. Surgery is done to treat bone disorder, especially any other disorder in our bones, accident-related concern, age, or something like that. So that's what we mean by orthopedic surgery. The market share of reconstruction joint is the highest in comparison to the other implant. Implant might be also dental implant. Dental is also an important branch in medicine. The main is joint, reconstruction joint, which means osteoporosis or osteoarthritis. Osteoporosis, that means weakening of bone. The bone becomes fragile and could lead to fracture. So that's osteoporosis that many people need to include or to make some kinds of surgery to repair bone. Osteoarthritis is kinds of degeneration joint cartilage that makes some pain in knee, hip, or something like that, some disorder. These are medical expressions. The orthopedic biomaterial market is one of the most important market and hold a significant share. We have material type like ceramic, bioactive glasses, metals, all these materials. Application for implant, some screw, some plates that we have to insert in the human body. So all these are for applications by using this type of material. End user is also hospital and clinics. The market in 2017 is 45 billion, expected to reach more than 65 billion in 2025. The role of orthopedic implant is to replace or provide support to damaged bone or joints. That's why we are using implant to heal, to cure bone disorder. What do we mean by biomaterial? All material intended for application in humans are biomaterials. As biomaterials or medical device, when you implanted these kinds of biomaterial in the human body, some kinds of tissue response to this material. Bio, that's mean for human being. Okay. According to the response of the tissue or the body to this implant, we have to classify what is the type of our material. If the tissue dies, this biomaterial is toxic. We don't have to use it. If it is inert, that means it forms fibrous tissue. If this biomaterial make a kind of link with our bone, it is considered bioactive, that have a certain bioactivity, and it's good. If it promotes some kinds of replacement, like something of disorder, something like that, the material is considered soluble. That's the response of biomaterial if we insert it in human body. Bone, and this has a kind of calcium phosphate. It's hydroxyapatite. Hydroxyapatite is a form of calcium phosphate. Bone is like a building. Bone provides shape for us, provides support for our body. We are standing like that because of our bone. So bone is very important for human being. Even make some protection for our organs, brains, stomach, whatever. So bone is a protector for us and also is a storage site for mineral. A material to be used as a biomaterial there is some kinds of characteristics. What kinds of biomaterial? Is it biocompatible? That means it's good, no toxicity, biocompatible with human body. Biofunctional, could have biofunctionality. That depends on what we are planning to do. This material is bioactive, bioinert. Bioactive, that means some kinds of reaction with the human plasma and generate the kinds of calcium phosphates. So we have to know from the beginning the characteristics of our material. When we used, or when we say biodegradable metal is magnesium, iron, zinc, calcium. Biodegradable, that means are intended to degrade in vivo, degrade in our body, and can be completely dissolved after completion of their function for tissue growth, which means full degradation of this implant. This implant might be magnesium, iron, it's okay, zinc, it's okay, calcium, it's okay also. Second, this degradation can be cleared by our metabolism, so no toxicity at all. And in comparison to polymer, these kinds of biomaterial metals have better mechanical properties than polymer. That's to get information about what do you mean by in vivo and in vitro. In vivo, that means inside the body. In vitro, outside the body. So to know when we say in vivo or in vitro. It be also in animals. So metallic biomaterial. We have some common metals and alloys that we are still using in orthopedic surgery. Stainless steel, cobalt-based alloy, or titanium alloy. Stainless steel and titanium are widely used in the medical implants. They're using as implant in the medical field. Why? Bioinert, mechanical properties, good mechanical properties, biocompatible. Like, for example, in our kitchen top, we are using stainless steel. Why stainless steel? Corrosion resistance. Cannot be corroded. They are using water, oil, whatever, what kinds of materials. Still, stainless steel resist. So, till now, they are using titanium alloy and stainless steel in surgery. In comparison to magnesium, magnesium is light, soft, has good mechanical properties similar to our bone, almost the same. But the problem of magnesium is degradation rate. It's very fast degradable once inserted in human body because it's very reactive. And that's, we have to stop this reactivity to make a good recovery after surgery. So sometimes they're using some kinds of alloy to increase the corrosion resistance of magnesium and to delay its corrosion also. But still now, no concrete results about the coating to resist the corrosion. That's a table about the difference between metallic biomaterial and magnesium. Stainless steel and titanium alloy have good corrosion resistance. And stainless steel wear resistance. Wear resistance that mean cannot be destroyed by friction or use. That's why we are using stainless steel in our kitchen top. Cannot be destroyed. by friction or use, you can use it for more than 10 years, no problem. But the problem of titanium alloy and stainless steel and even cobalt alloy is lack of biodegradability. They cannot, it's toxic. That's a problem. Sometimes they have to do another surgery to get it out. So in comparison to magnesium, biodegradable, biocompatible, mechanical properties similar to those of bone. So it's perfect. But the problem of magnesium is its low corrosion resistance. This low corrosion resistance lead to hydrogen bubble, and this hydrogen bubble after that some kinds of dissolution and make a product is magnesium hydroxide, which increase the level of alkalization around the tissue, and that's it's not good. In a quick comparison, why magnesium and bone and others? Just the density. If we choose the density as the parameters to make a comparison, bone and magnesium almost same density. The other heavy metal, titanium, iron, nickel, and even cobalt, and all heavy. That's mean if we insert an implant in the body, there is a kind of mismatch between bone and the implant, and make later implant failure because there is no mismatch between the two. What is corrosion? Corrosion is the degradation of the material properties. That means the material properties due to exposure to environment, whatever it's acids or even physiological environment, the material degrade, their properties degrade. And that's it's a very important parameter. That's why when we do corrosion, we have to do some kinds of characterization in terms of corrosion resistance of this material or the coating itself. When we are doing some kinds, or we are using some kinds of medical implant design, we have first of all to know the physico-chemical properties of this material. In our case, it's magnesium. We have to know its physical chemical properties, which dictate after that the mechanical properties, and later what kinds of coating we are planning to use, to apply for these kinds of substrate or implant, because after that will lead to informations about our desired cellular response, what kinds of cellular response we are planning to get. So we have to have all this information, physical properties, mechanical properties, topography of this coat, it's okay or not, it's completely covered the metal or the substrate completely or not. That's all this we have to take into consideration when we are using magnesium special. That's also a kind of a small comparison between mechanical properties of the widely used, we call stainless steel and titanium in comparison to bone. The first impression just to the mechanical properties is completely too high in comparison to bone. And that's many times they have to do another operation to get these kinds of implant out because of mismatching of properties. Something like you are doing a load, you are putting the titanium or stainless steel over a bone, which is very light, so some kinds of pressure, and that's lead to failure later. But the only material that's much completely the properties of bones is magnesium. Why magnesium should be protected? That's why we have to use coating to protect these kinds of implants. Magnesium, just put in any other solutions, is completely dissolved. We make ions, and then finally you get magnesium hydroxide, which increases the alkalization and hydrogen bubble. The idea is to prevent this reaction. In our body, all of us use magnesium. That's why no problem. Magnesium can be dissolved, no toxicity, because every human being needs calcium, magnesium, zinc, and iron also. That's the quantity that all of us needs for our body and our system. So it's okay to use magnesium, even magnesium alloy with calcium and zinc. It's okay, no problem. But the problem is the corrosion of magnesium. The human plasma contains all these kinds of ions. Our temperature is almost 37 degrees centigrade. All this ion, the one which is the most aggressive is chloride ion. That's the ion which completely degrade or destroy magnesium. That's illustration of all corrosion reaction. When we magnesium, you have a lot. We have enzyme, we have proteins in our body, and all these kinds of reactions. That's completely destroyed. It's really complex corrosion scenario. It's not just like the reaction in magnesium and get magnesium hydroxide and hydrogen. But no. It's very complicated chemical reactions. So that's completely destroy the magnesium. That's a small illustration, just to know how it comes. That's the magnesium substrate. Is the first here, okay. Some kinds of dissolution and react. Bubble, and also react with phosphate to give magnesium hydrogen phosphate. It's a kinds of layer which protect corrosion. But come chloride ion to destroy completely this layer, because chlor is a very aggressive. So we do have magnesium, right? And like here, okay, it's corrosion of magnesium completely corroded completely. So you cannot use magnesium in itself. We have to make a kinds of coating to protect it. To protect magnesium, we do have some kinds of surface modification. We have to modify the surface of magnesium. We have to know the chemistry of the coating we are planning to apply on this substrate to make some kinds of roughening of the surface. Roughening, that means I am preparing the surface of magnesium to be coated by the composite. That's roughening. And after that, to make a kind of deposition of this surface. The third material we are still using in our work is bioactive ceramic. We have two bioactive ceramics, hydroxyapatite and bioglass. Why? Both form a bond, like a link between the living tissue and the material. Once inserted in vivo or in vitro, some kinds of bond, some kinds of cell profilation, some kinds of stimulation to make a apatite on the surface. So that's why they are bioactive ceramic. They are added to any type of coating to improve the bioactivity. That's the composition of bioglass. Should be, you have calcium, you have sodium, you have especially phosphorus oxide, you have silica. That's all type of bioglass, which it's still many use, for example, apatite, wollastonite, glasses that depend also type of bioactivity. In our work here, we are using bioglass. The reaction stage of this bioglass is very complicated in vivo. You have a lot of stages of reactions, from ion exchange to dissolution to attachment to nucleations to differentiation to cellular attachment. So it's not like put something, it's not easy. You have to follow all these kinds of reactions. Just to give you information about the sequence of reactions that's happening when we put an implant in human body. That's now we are moving to our work of Preceramic Organosilicon Polymer, which the polymer we are using in our work is at Hermes. We are using these kinds of synthetic polymer. We are using this synthetic polymer with another polymer, which is chitosan, as natural polymer. So we are using two kinds or two types of polymer. And we are using bioglass as a bioactive material to promote bioactivity. Our objective is to make a good corrosion-resistant coating to protect magnesium from corrosion. That's the most important point. And after that, to discuss the cellular response to our coating. What would be the situation? Do we have some kinds of proliferation, some tissue growth? These kinds of coating, for example, would destroy the cells. We have to study all these things. The problem is we want to reach a decreased rate of biodegradability of magnesium to give time to our body to form a kind of calcium phosphate, or to make this link between bones, because we are using implant to heal or to cure some kinds of fracture or to treat some kinds of bone disorder. So we have to give time with this coating for the magnesium to recover and resist the human plasma. Organosilicon polymer has this formula. According to the X, you could get polysilane, or polycarbosilane, polysiloxane, or polysilazane. Our work is just on polysiloxane and polysilazane. Polysiloxane is SiOC, and polysilazane is SiCN carbonitrate. That's the route to prepare or to get to this ceramic. The main functionality of this polymer-derived ceramic is you are starting by polymer and finish by ceramic. So you have almost composite. So this one of the most important functionality of this Preceramic Organosilicon Polymer. It's a polymer, starting as a polymer, but after certain treatment, or controlling condition, you could get ceramic. First of all, that's a molecular precursor, some kinds of polymerization, and we got here pre-ceramic polymer. That's our work. We start from pre-ceramic polymer after getting the material from the company. Pre-ceramic polymer could be shipped. For example, like any polymer technology, shipping could be casting, could be injection, could be extrusion, could be even by additive manufacturing, could use laser, whatever, tape casting. Even we could make fiber, because it's a polymer, you could make fiber from it. Casting or impregnation. Our work here is casting, is coating. We are using coating as a shaping technology. That's first. Second, we have to do cross-linking. Cross-linking, I would explain it in a simple manner. For example, you have a plate of spaghetti. Okay? Each strand of it, it's alone to get it. Okay? Cross-linking is making all this spaghetti together as one unit. That's cross-linking. Polymer is some kinds of repeating unit. You have many repeating units of polymer. For example, this N, this polymer have repeating unit of this. That's why we make cross-linking to make them together as one unit instead of many units, and to be very hard to resist any other corrosion or any other atmosphere. And then we do some kinds of pyrolysis, if you want, okay, to get the final SiOC, silicon oxycarbide or silicon carbonitride. This material has been used till now for industrial application, not for bio application. Not too much work has been done till now on the application of this organosilicon polymer for biomaterial. Just you could count the number of research on your fingers, but some research, it's okay. It's a biocompatible material that could be used to protect, because they have a lot of interesting properties, including corrosion resistance, oxidation and thermal resistance. So has a lot of we call fabulous properties. They are using silicon-based implant. Silicon, it's polysiloxane. Polysiloxane means silicones. So it's used in the medicine for a variety of application, ranging from total replacement of organs and tissue, food replacement, sometimes many other replacement, other for corrective or repair device, like pacemaker, for example, tubing for heart valve. They are using silicone till now for those, and that's illustration for placement of the silicone-based implant and device in the human body. A lot of applications, even for wound dressing, catheters also. So till now, a lot of application for silicone-based implant. It's biocompatible. That's some of my work that I did in Germany, for example, some coating, but on stainless steel. I used polysiloxane, and we did some kinds of cytotoxicity to get information about this material is toxic or not. We have to do some kinds of test. It's okay. The cells didn't die at all, so could be used for coating and for implant. When we are doing coating, we have to do some kinds of characterization. Characterization, especially surface characterization, and make some kinds of analysis, also chemical analysis when we are using SBF. SBF, that means silicon simulated body fluids. Like our plasma, we are using the same composition of our plasma, which contain chloride, bicarbonate, sodium, calcium, phosphate, and everything, and make it by using pure materials to make our test. We call it in vitro, now in vitro. Surface characterization, we use HBSS, which is very important tool to get information about what kinds of coating are you, and give every element, the analysis of every element on the surface. XPS, XRD, sometimes you have to use XRD, X-ray diffraction, FTIR, that means infrared, SEM, that means scanning electron microscope, EDX, we have to, EDX, that means when you are looking under the microscope and you have some grains, I will show it later with the next slides, to make on your samples some X-ray under microscope to get information about what type of grain you have. TEM is transmission electron microscope, which is for nanomaterials. Sometimes if you have nano product or you are using nanomaterials in your coating, you have to do some kinds of transmission, which is a higher degree of microscope. Atomic force microscopy is also very important to get information about sometimes thickness or thickness of the coat itself you are using, or give you information about the hardness of the coating. Is it hard coating or not? That's atomic force microscopy, which is also very important tools. And EIS is electrochemical impedance spectroscopy. That's to measure the corrosion resistance of your coat. EIS. We did FTIR to get information about, we call it the link we have after coating, and that gives indication. For example, that's a little bit easier, this one. That's the pure chitosan we are using. Okay. After we call conversion coating with magnesium, they form a complex. That means some kinds of reaction has happened between magnesium and pure chitosan. So that gives here indications that some kinds of change has happened in our coating structure. That's called potentiodynamic polarization or electrochemical impedance. It's a little bit complicated, but that gives indications about the resistance of your coat. And this type of equipment give you indications about also corrosion rate. The corrosion rate, we have to make some kinds of tangent, this polarization, and that's the impedance. The right one, it's impedance. Okay, there is 2 peak, and that's give indications about how big is the resistance to your coat. That's a little bit complicated. That's here give you indications about corrosion rate, which is we reach about 0.04 milli per year. That's we call the coating morphology. That's the big under microscope. We are seeing this kind, so you have to, it's completely covered the surface of our magnesium. And then here we have to, if I need to get information about, for example, this small here, what about the composition of this small zone or area? That's by edX. That's inside the microscope. We have to use X-ray to get information about what kinds of coat we have in this area. That's your measure we have magnesium. But that's the darker area. If we go to a lighter area, you have here some kind of 34% magnesium, and you have the ultimate position of the magnesium. And that's the analysis, quantitative analysis. And that's we call the chart that gives you indications about all the elements. Don't know, it's a little bit, but if you make zoom in this area, we got this. So, for example, okay. So that's analysis. And we go more to do also to get information about these small grains and its shape, and also this analysis. So that we do indications about what kinds of grain here, what kinds of grains there, and which is very important to control your coating process. And that's XPS. XPS, it's just for surface. If you want to make any analysis for surface, you have to use XPS. XPS, you have this coat. We have to know what kinds of material I do have. Calcium, have to calcium. Okay. Phosphorus, okay. What do I have again? Boron, do I have nitrogen, whatever. And XPS makes the analysis of the surface. The analysis of the surface is just between 1 and 3 nanometer, which means 2 to the power minus 6 millimeter. This beam go exactly to 10 to the minus 0.6 millimeter to make the analysis of the surface. And that gives you indication what do you have exactly on the surface with all the compositions, the percentage, the quantity of this element that present in your coat. Which means also here that, for example, if we use this calcium, which is a strong here, that means you do have, for example, 2 types of calcium. That means 2 types of compound. Perhaps tricalcium phosphate or dicalcium phosphate. So, according to the P, we could expect what kinds of product you have. And finally, the field of biomaterials is interdisciplinary field. This means it's you have to make a kinds of working together of big team of material engineering. First of all, you need mechanical engineering to test some kinds of mechanical properties. You need the chemist to make some kinds of chemical analysis after using simulated body fluid. You need the biology to make some kinds of cell culture. We need a doctor, for example, to make this implant and test the in vivo behavior of your implant. So it's interdisciplinary area, these biomaterials. You are working with all kinds of disciplines and branches. And finally, thank you for your attention.